The people living past 100, the centenarians we study, the ones who age without the diseases most adults expect, they show one biological feature that almost no one in the health space talks about seriously. It is not their genetics, it is not their supplements. It is what is living inside their gut. Specifically, what is diverse inside their gut. Some of you have a gut ecosystem right now that resembles a monoculture field. One or two dominant species, very little variety, and it is shaping your inflammation, your metabolism, your cognitive clarity, and your immune response every single day without your awareness. Today, we change that. I'm Dr. Elena Churkina, lifestyle medicine advocate. This channel is dedicated to evidence-based strategies for busy adults who want to prevent disease, stay cognitively sharp, and age with intention. In the next 12 minutes, I will give you a 7-step evidence-based microbiome diversity protocol, a 4-week implementation plan, and, crucially, one biological pathway most practitioners do not include. The role of animal contact and outdoor exposure in rebuilding your microbial ecosystem from the outside in. Stay with me. Your gut microbiome is a community, bacteria, fungi, viruses, and other microbes living primarily in your large intestine. They digest fiber. They produce short-chain fatty acids. They train your immune system, influence systemic inflammation, support metabolic function, and communicate directly with your brain via the gut-brain axis. Modern healthy adults typically carry between 200 and 300 bacteria species in the gut. That number sounds large. It is not, compared to what the human microbiome can sustain. Centenarian studies consistently show one finding: people who age to 100 and beyond tend to carry significantly higher microbial richness and diversity compared with younger, less healthy older adults. The microbiome show more Akkermansia, more lactobacillus, more Rosbifera, bacteria specifically linked to butyrate production, immune regulation, and metabolic resilience. I want to be precise here. We cannot say centenarians have exactly 400 or 500 species. Study methodology, testing methods, geography, and individual variation all affect those numbers. What the research consistently shows is this: healthy aging is associated with a richer, more flexible, more metabolically active microbiome. The lesson is not to chase a number. The lesson is to build an internal ecosystem that is diverse, resilient, and functional. Not because you like discipline, but because your current food environment, stress load, and lifestyle have been quietly reducing that diversity for years. And no one gave you a protocol to reverse that. Until now. Step 1. Eat 30 different plant foods per week. Not 30 servings of the same 5 vegetables. 30 distinct varieties. Because different plants feed different bacteria. Your gut microbiome has dietary preferences that are as individual and specific as yours. Count everything: vegetables, fruits, beans, lentils, chickpeas, whole grains, nuts, seeds, herbs, and spices. A single Monday can yield 12 or 15 varieties: oats, chia, berries, walnuts at breakfast, lentils, spinach, carrot, onions at lunch, brown rice, broccoli, garlic, mushrooms at dinner. Most people try to improve gut health by editing one probiotic capsule. The real mechanism is not one capsule. It is the ecosystem you feed every single day. Step 2. Increase fiber slowly and strategically. Fiber is a primary fuel for beneficial bacteria. Its fermentation produces short-chain fatty acids, butyrates, acetate, propionate, which support the gut lining, regulate immune signaling, and protect metabolic function. Your target for most adults is 25 to 35 grams per day. Most of you are not currently at half that. Do not attempt a sudden increase. Jumping from low to high fiber overnight produces bloating, gas, and significant discomfort, which causes people to abandon the protocol entirely. Week 1, add 1 extra plant portion daily. Week 2, add legumes 3 times per week. Week 3, add seeds daily, chia, flax, pumpkin, or sesame. Week 4, introduce resistant starch, cooked potatoes, cooked rice, green banana flour, or beans. Step 3, add fermented foods. Start it very small. A Stanford randomized trial found that 10 weeks of fermented food-rich diet measurably increased microbiome diversity and reduced inflammatory markers in participants. The options: kefir, live yogurt, sauerkraut, kimchi, miso, tempeh, kombucha, and traditionally fermented pickles. Begin with 1 teaspoon of sauerkraut or few tablespoons of kefir or 1 serving of live yogurt. If fermented foods trigger significant symptoms, slow down and personalize. Your gut protocol must match your gut physiology. Step 4. Feed Akkermansia and your Butyrate producers. Imagine your gut lining as a garden wall. Akkermansia is one of primary microbes that maintains that wall. Centenarian microbiomes consistently show elevated Akkermansia levels. To support Akkermansia, polyphenol-rich foods, pomegranate, cranberries, blueberries, green tea, cacao, and colorful plant varieties. To support Butyrate producers, oats, barley, beans, lentils, onion, garlic, asparagus, leeks, apples, ground flaxseed, and resistant starch. Food first. Always food first. Step 5. Reduce your microbiome disruptors. The main disruptors are unnecessary antibiotics, ultra-processed food, alcohol, high added sugar, chronic physiological stress, poor sleep, low movement, and, critically, a repetitive diet. You cannot build diversity by eating the same 10 foods every week. Rotation is a biological requirement. Simple swaps: white bread daily to rye, oats, sourdough, or mixed grains. Sweet snacks to fruit, nuts, dark chocolate, live yogurt. Same breakfast every day to a rotation of 3. No movement to a 10-minute walk after meals. This is not about perfection. It is about reducing frequency. Step 6. Move consistently. If you watched our lectures on sedentary lifestyle and microbiodiversity, you already know the mechanism. Exercise directly influences microbial composition, gut motility, inflammation, and metabolic function. 2 strength sessions per week, 10 minutes walking after 1 or 2 meals every single day. 1 longer nature walk per week. Consistency beats intensity, always. Step 7. Protect your circadian rhythm. Your gut microbes follow rhythms. Irregular eating schedules, late night snacking, and fragmented sleep disrupt the microbial environment independently of what you eat. A consistent sleep-wake time, a 12-hour overnight window between dinner and breakfast where possible. No heavy meals immediately before bed. Morning light within 30 minutes of waking. These are not optional lifestyle preferences. These are biological inputs your microbiome requires to function. Now, let's talk about the pathway almost no gut health protocol includes. You can eat every fermented food on the list, hit 30 plant varieties, and optimize your fiber perfectly, and still be missing one of the most ancient and biological significant routes to microbiodiversity available to you. Contact with animals. Contact with soil. contact with a living world outside your front door. A study published in Frontiers in Cellular and Infectious Microbiology found that pet ownership, specifically dogs' ownership, was associated with measurably different gut microbiota composition compared to households without pets. Dog owners and their dogs show significantly overlapping microbial profiles, sharing the same primary phyla and families. The mechanism is not complicated. Dogs bring outdoor microbes into your home. You share physical contact. You co-inhibit environments. You exchange microbiome information constantly in a way that your immune system was evolutionarily designed to use. A 2025 study confirmed that dog ownership during adolescence measurably alters microbiota composition. It is not specifically dogs. It is microbial exposure for living systems, animal skin, outdoor soil, plant surfaces that your human architecture was designed to interact with. We evolve in constant contact with diverse microbial environments. Modern, sanitized, climate-controlled life has removed the majority of that contact, and your microbiome reflects that removal. Soil and plant contact compounds this further. A 2018 study published in Microbiology Open found a pronounced increase in skin microbiota diversity immediately after short-term exposure of hands to natural soil and plant material during gardening. A 2022 study in Scientific Reports found that soil-delivered bacteria were detected in human fecal samples and increased with direct and indirect soil contact. Now, let me tell you where almost everyone fails at this protocol. They do it for 2 weeks. They feel better. They stop tracking because tracking feels like effort. And within 8 weeks, they are back to 6 plant foods per week, no fermented foods, and 0 outdoor contact. A diverse microbiome is not built in one day. It is trained like a muscle, and like a muscle, it requires consistent load to maintain its capacity. The moment you stop loading diversity inputs, food variety, fermented sources, outdoor exposure, animal contact, your microbial richness begins to contract again. This is not a 4-week protocol. This is a permanent architecture change in how you relate to your food environment and your natural environment. A diverse microbiome is not a wellness trend. It is your biological inheritance, shaped by millions of years of contact with food, animals, soil, and the living world around you. The evidence is clear. The protocol is specific. The only variable is you implement it over clinical interventions. I'm Dr. Elena Chirkina, evidence-based medicine for the life you are actually living. I will see you in my next lecture.